Hello students, this is your instructor Abid Jan, and today's lecture is about protein sequence databases. The protein information resource. The protein sequence database was developed at the National Biomedical Research Foundation in the early 1960s by Margaret A. Hoff as a collection of sequences for investigating evolutionary relationships among proteins. Since 1988, the protein sequencing database has been maintained collaboratively by PIR International, an association of macromolecular sequence data collection centers. The consortium includes the Protein Information Resource at the NBRF, the International Protein Information Database of Japan, and the Martin Street Institute of Protein Sequencing. Different sections in PIR. In its current form, the database is split into 4 distinct sections designated from PIR 1 to PIR 4, which differ in terms of the quality of data and level of annotation provided. PIR contains fully classified and annotated entries. PIR 2 includes preliminary entries which have not been thoroughly reviewed and therefore may contain redundancy. PIR 3 contains unverified entries which have not been reviewed. PIR 4 entries fall into one of 4 categories: conceptual translations of artifactual sequences, conceptual translation of sequences that are not transcribed or translated, protein sequences that are extensively genetically engineered, sequences that are not genetically encoded and not produced on ribosomes. Another protein sequence database is SwissProt. SwissProt is a protein sequence database. The database endeavors to provide high-level annotations, including description of the function of protein and of the structure of its domains, its post-translational modifications, variants, and so on. And SwissProt aims to be minimally redundant. The structure of SwissProt entries. The database contained 562,755 entries from more than 5,000 different species, and the bulk of these coming from just a small number of model organisms, for example Homo sapiens, Saccharomyces cerevisiae, Escherichia coli, Musculus, and Rotus norvegicus. Each entry corresponds to a single sequence as contributed to the database. Each line is flagged with a 2-letter code which helps to present the information in a structured way. Entries begin with an identification line and end with a terminator. ID codes in Swissprot have been designed to be informative and user-friendly. They take the form protein-source, where the protein part of the code is an abbreviation that denotes the type of protein, and source indicates the organism name. For example, we can have INS-human, so INS insulin protein from human. Swissprot lines. The accession number is provided on the AC line. Although relatively uninformative to the human user, it is, however, computer readable. If several numbers appear on the same accession line, the first or primary accession number is the most current. Next, the DT line provides information about the date of entry of the sequence to the database and details of when it was last modified. The description DE line informs us of the name by which the protein is known. The following lines give the gene name, the organism species, and organism classification within the biological kingdoms. Supporting references. The next section of the database provides a list of supporting references. These can be from the literature, unpublished information submitted directly from sequencing projects, and data from structural or mutagenesis studies. Therefore, the database is thus an important repository of information that is difficult or impossible to find elsewhere. The comment lines following the references are found comment lines. These are divided into themes which tell us about the function of the protein, its post-translational modification, its tissue specificity, and subcellular location. The DR and FT lines. Database cross-reference lines, DR lines, follow the comment field. DR lines provide links to other biomolecular databases, including primary sources, secondary databases, and specialized databases. After the DR lines are found a number of FT lines which form what is known as a feature table. The feature table highlights regions of interest in the sequence, including local secondary structure, ligand binding site, post-translational modification. The SQ lines are sequence lines. The final section of database entry includes the sequence itself on the SQ lines. For efficiency of storage, the single letter amino acid code is used, each line containing 60 residues. Sequence data in Swissprot correspond to the precursor form of the protein before post-translational processing. Hence, information concerning the size of molecular weight will not be necessarily correspond to values for the mature protein. This is the bibliography about the lecture. Stay calm. Allah Hafiz.